It's absolutely a fantastic time to be interested in fungi. In terms of genetics, what we have, which we didn't have just a few years ago, for example, were genome sequences. We have genome sequences now for any number of fungi and more on the way. We have gene transfer methods, so we can manipulate the genomes for research purposes anyway, very useful. So I don't think there's ever been a better time to work with fungi. So our project is part of the sort of broader remit of LACE. So the aim of LACE is to convert plant cell wall polymers to ethanol, but to start with waste organic matter. So we're not competing with foodstuffs. So there are basically three steps. You've got to open up the plant cell walls to make those more accessible to enzymes. We're involved in the next step where we use enzymes to convert the plant cell wall polymers to monomers.
So our task within our strand of the project is to break down complex polysaccharides such as cellulose and hemicellulose and make them into simple sugars such as glucose and xylose. I'll just give an example of what we're talking about here. What I have here is a flask of pure glucose. This is glucose in its monomeric form, and you can see it's nice and clear solution as it's completely soluble. And that's what we want to use to convert into ethanol during fermentation. So what we have present in plant cell walls, such as in the straw that we use in this project, is complex polysaccharides, such as this one here is cellulose. And all the cellulose is simply a long chain of glucose molecules simply bound together. And you can see that simply by binding the glucose molecules together into a polysaccharide and adding no other residues, we've created something that's actually quite insoluble, and we get sort of a powder suspending liquid here that won't go into solution. And there's actually exactly the same amount of glucose molecules in this solution as there is in this one, but just simply because of the polysaccharide nature of it, you can see that it becomes very insoluble, and this is quite difficult to degrade, and that's what we're trying to do in our project.
So to doing this, we use fungi. So here it's an example of some fungi who grow on a plate. And so fungi, what is the most famous part that people know is the mushroom out of it, but what we use here of this fungi, it's used because it's widespread in nature, and all the fungi have the capacity and contain a lot of enzymes which are susceptible to degrade the plant cell wall, such as straw, the substrate we use. And so we use a lot of tools to understand how the fungi are able to degrade the plant cell wall. And so for that we do some genetics, and here I can present you two mutants. So the fungi we use is called Aspergillus niger, and here it's a wild type, and you can see is totally clear and white, but using genetics we can create some mutant to understand how this fungi is able to degrade plant cell wall, and some mutant make a funny color like this one, a bright yellow. And so using genetics, biochemistry, cellular biology, we try to understand how these fungi are able to go from this cellulose, this complex carbohydrate, to produce just proper liquid translucid sugar, single monomer.
And then the final step is to use yeast to convert glucose to ethanol. So we're in the middle using enzymes from fungi. Well, I would have strong interface anyway with the yeast people, Catherine and Ed. So I think that would have happened anyway, but the more social and ethical aspects we wouldn't have interfaced with at all in my group, but now I'm quite pleased that we are. So yeah, it is a large project, and we're pleased to be part of it, for sure. Yeah.